video provides a brief overview of adaptive immunity. Remember that our adaptive immune system has two arms, one that provides humoral immunity and one that provides cellular immunity. Cellular immunity is orchestrated by our T lymphocytes. We have T cells that can activate other T cells and B cells, such as our helper CD4 T cells. We also have T cells, like our CD8 cytotoxic T cells, that can control intracellular viral infections, among other things. As you may have noted, we can identify T cells through specific markers on their cell surface called cluster of differentiation, or CD molecules. Our helper T cells are often identified as CD4 T cells, while our cytotoxic T cells are often identified as CD8. You may see CD25 T cells, and these are the regulatory T cells that help dampen the immune response. Our CD4 helper T cells are master regulators of the immune system. Helper T cells are required to present a robust adaptive immune response. CD4 helper T cells are able to communicate with MHC2 molecules on the surface of antigen-presenting cells, like the dendritic cells seen in this picture. These antigen-presenting cells present antigen to the helper T cell, which then activates the helper T cell, so it can then go direct other cells to respond to this antigen using chemical signals called cytokines. Our CD8 cytotoxic T lymphocytes are able to recognize antigen on MHC1 molecules. By communicating with a foreign antigen on MHC1 molecules, CD8 T cells are able to recognize and destroy cells infected with a virus, cells that are infected with intracellular bacteria, such as mycobacterium species that cause tuberculosis, and cancer cells. In addition, allergens presented as antigens on MHC1 molecules can direct an allergic or hypersensitivity response through a CD8 cytotoxic T cell. Cytotoxic T lymphocytes can destroy target cells through releasing cytotoxic enzymes that can damage the plasma membranes of cells, such as perforins, and through triggering apoptosis through a death receptor on the surface of the cell. Both of these mechanisms will remove an infected cell from the body, preventing further replication and further disease. Remember that our immune cells will have receptors that attach to these MHC molecules and are able to recognize the antigen. On our T cells, these are called T cell receptors. And in our B cells, they're called B cell receptors. These are important because only those cells whose receptor, like the T cell receptor, can fit the antigen that is being displayed by that MHC molecule is able to respond to it. This is the specificity that our adaptive immune cells have. The antigen-presenting cell can present any antigen that it finds and recognizes as foreign. But it has to be able to find a T cell whose T cell receptor is able to specifically fit the antigen that antigen-presenting cell is presenting. So I like to think of it as the key master and the gatekeeper. You can have an antigen-presenting cell and it's going to present its antigen to a bunch of, let's say, helper T cells, but until it finds the right helper T cell that can fit that antigen, it's not going to create that immune response. Our humoral immunity is mediated through B lymphocytes. In humoral immunity, our B cells function to eliminate extracellular microbes and toxins. These are key, as creating memory will lead us to a heightened response if there is a subsequent exposure. activation of helper T cells, as you can see in this picture here, through binding of the antigen-presenting cell and some cytokine signaling, will then initiate and activate a helper T cell to go find some B cells and say, hey, this is this antigen that we found. I need you to make some antibodies for it. By talking to the B cell, the helper T cell will activate this B cell, causing it to proliferate or increase in number. A B cell can turn into a plasma cell through the process of differentiation, where it becomes a little antibody factory, or a B cell could differentiate into a memory B cell, where it hangs out for a long time, ready to proliferate and make more plasma cells with subsequent infection. When we have a primary immune response, our B cells and T cells are naive. Let's look at this from the humoral antibody immunity perspective. During initial exposure, or the primary immune response, an antigen is presented on an MHC2 molecule by an antigen-presenting cell. It then goes and activates a helper T cell, which then goes and talks to its friends, the B cell, causing these cells to proliferate or divide and increase in number, and differentiate or become more specialized, activated, and eventually turn into a plasma cell. They can also opt to turn into a memory B cell so that you save some response for later. In the primary immune response, the plasma cells will produce antibodies. And so you can see where there's a delay, where until after about 2 weeks, you don't start to get a rise in antibody numbers in the blood. But somewhere around week 3.5 or 4, you have a robust antibody response that can then start to resolve that infection. When you get a vaccination for the first time, this also produces a primary immune response. Whether you get the disease or you get a vaccination, you will create a memory B cell that hangs around and waits. So the next time we see this antigen, we get a fast and specific secondary immune response. The secondary immune response occurs when there's a subsequent exposure to antigens. Because we have memory B cells, plasma antibodies levels are able to rise within days because they can respond immediately. You get this secondary response if you get reinfected with a disease you've had recently or previously, or when you get booster shots where you are trying to stimulate a higher level of antibody before a disease is encountered. So the initiation of the adaptive immune response is where the innate and the adaptive immune systems meet. Our antigen presentation occurs primarily through macrophages and dendritic cells, which are part of the immune system that is innate. Macrophages and dendritic cells are not specific, but they do act as signals to help activate the immune response by talking to our helper T cells. To initiate an adaptive immune response, you need 2 signals. The first is you need an antigen presented through an MHC2 molecule. Secondly, your antigen-presenting cell is going to release some signaling molecules called cytokines that activate the helper T cell and says, hey, okay, I've showed you this antigen, now let's get to work and get rid of it. So let's go through the basic steps of an adaptive immune response. The first step is that an antigen-presenting cell ingests the antigen. Our primary antigen-presenting cells are macrophages and dendritic cells. But remember that B cells can also serve as an antigen-presenting cell. Antigen-presenting cells are able to ingest the antigen and process it into smaller pieces. antigen-presenting cells also have MHC2 molecules on their surface. During the processing, antigen-presenting cells will connect an MHC2 molecule to the antigen that they've processed and bring that up to the surface. Once the antigen-presenting cell has brought this MHC2 molecule with the antigen to the surface, it then goes finds a CD4 helper T cell that has a T cell receptor that can recognize this antigen. Once it's found its buddy, it then sends these proteins called cytokines that signal the T cell, hey, let's get activated. So when you have this antigen presentation plus the cytokine signaling, we now have an activated helper T cell. Once activated, our helper T cell then communicates with our B cells and our CD8 cytotoxic T cells. When the CD4 helper T cell talks to the cytotoxic T cell, it then tells the T cell to become active. You're now an effector cell that can eliminate foreign antigens by killing any cell who displays that antigen on its MHC1 molecule, so any infected cell or a cancer cell. Some of our cytotoxic T cells also become memory T cells because we want to have those for later. Our helper T cells also talk to our B cells. By binding to the B cell and sending some cytokines over, the B cell is like, oh, hey, let me respond to this antigen. Remember, B cells respond to extracellular pathogens. So they can differentiate into a plasma cell, which makes antibodies that can circulate through our blood, in our body fluids, or live in our mucous membranes, capturing any antigen that it recognizes based on its priming from earlier. Some B cells also differentiate into memory B cells, so they can hang out and be ready for later. So let's summarize this quickly. The 4 steps of the adaptive immune response are: 1, an antigen-presenting cell will ingest the antigen, process it, pair it with an MHC2 molecule, and then bring it to the surface and present it to the T cell receptor of a helper T cell. This activates the helper T cell. Once activated, the helper T cell then goes and talks to a B cell and to the CD8 cytotoxic T lymphocytes.